Hello my dear aspirants, I hope you all are doing well. I hope your preparations for CSR NEET exam is going very well. So as you don't have much time left for your exam, so it's high time for revision, practice and going through all the topics that you have once studied. So today I am back with another very important topic. Actually, I am here with a series of immunology important topics. As you all know that immunology is a very important part for your CSIR NEET exam. The important topics of immunology are innate and adaptive immunity, then your MHC molecules, structure of antibodies, complement system, B and T cell activation. So in this video, I'll be talking about innate and adaptive immunity, and I will talk about the MHC molecules. In our next video, I'll be discussing about complement system and other remaining portions. Watch the video till end, and if you like my videos, please share with your friends and please subscribe to my channel. And also, if you want to take detailed coaching of CSIR NET and Northeast Slate exam, then you can comment down below or you can contact in the given number. Immunology is the study of immune system, and what is immune system is a system of our body that helps to protect us from pathogens. And immune system can involve many different types of cells, molecules or organs. These are the list of some human pathogens and their the disease that are caused by these pathogens. You can take a screenshot for later. Here's another explanation of the immune system. Like, the immune system is a defense mechanism of our body that protects us from foreign non-self antigens with the help of immune cells. So these foreign molecules are the non-self. That means they are not the part of our body. Okay, they will come from maybe from outside, or they can cause harm. Those are called antigens. Like, so the immunity, it can be of two different types, innate and adaptive. So before going into this innate and adaptive immunity, which are the two different types of immunity, let's first discuss about the antigens. So this is important to know what are antigens. Let's first see the nature of antigen. What are antigens? Antigens can be a protein or a polysaccharide, nucleic acid or lipid. If it is a protein, then it will be highly immunogenic or highly antigenic, and if it is a lipid, then it will be poorly immunogenic. Now, what is immunogenicity and antigenicity? First you have to understand that also. See, antigens can be immunogens and antigens sometimes may not be immunogens. Fine. So it's not necessary that all antigens are immunogens. What are antigens? Any foreign material which are non-self molecules, they are antigens. But when an antigen becomes an immunogen, then only it can trigger the immune response. Fine. All antigens are not immunogens, but all immunogens are antigens. This word antigens, it actually describes about the foreignness of a molecule, how foreign it is. Sometimes a foreign molecule may not trigger any immune response. That means it may not activate our immune system if it is not necessary. If the antigen is immunogen, then only it will activate our immune system, and our immune system will work in order to eliminate that antigen. That means immunogens are harmful, but if an antigen is not immunogen, it may not be harmful. In simple words, you can understand that. Now coming to epitope. What are epitopes? Epitopes are the active region of an antigen. Suppose this is an antigen, and in the surface of an antigen there will be some pattern which will be identified by our immune system. This pattern will be called epitope, or you can call it antigenic determinants. In this epitope only our immune cells will bind. There are different types of epitopes. That means different antigens will have different types of epitope on their surface, which will be identified by the cells of our immune system. Antigens can be exogenous or endogenous. Exogenous antigens means from outside of the body, like the bacteria, virus, etc. Endogenous antigens means it will be from the inside of the body, like faulty protein, any defective protein, infected cells, etc., which can be harmful, which are not normal. Let's see the properties of antigens. So in order to be antigen, they have to be foreign. There should be foreignness. It should be a protein or polysaccharide, but not lipid, okay, because lipids are poorly immunogenic. Size, an ideal size of an antigen to become immunogen is 14 kilodalton to 600 kilodalton. Okay, this is a standard size. If it is smaller than this, then it is not an ideal antigen or an immunogen. Epitope should be present. Okay, epitope must be present. If there is no epitope, then where our immune cells will bind, right? So here is another explanation. If an antigen can trigger immune response, then it will be called immunogen. If antigens cannot trigger immune response, then it will be called non-immunogens, or you can call it heptin. Heptins have a very small molecular size, which is less than 14 kilodalton to 600 kilodalton, which we have already discussed that this is an ideal size. Now, if we want to make heptin an immunogen, then we have to add a carrier protein with it. If a carrier protein, or you can call it adjuvant, is added to heptin, then the heptin will become immunogen, and then it can trigger an immune response. What is antigenicity? The ability of a substance to bind to immune cells. And what is immunogenicity? The ability of a substance to bind to immune cells and generate immune response. If the molecule is antigen, then definitely it can bind to immune cell, but it cannot activate our immune response or the immune system. And if a molecule is immunogen, then it can bind to immune cells, and also it can generate or activate our immune system, or it can trigger our immune responses. Only heptin cannot trigger immune response, so no antibody will be formed. Triggering of immune response simply means formation of antibodies in our body. Heptin plus adjuvant. If heptin is mixed with the carrier protein adjuvant, then antibodies will be formed, and this you can call that our immune response is now triggered. Adjuvant only can also trigger the immune response, and thus only with adjuvant also antibodies will be formed. So here are some examples of heptin. You can take a screenshot and memorize later. Formation of immune cells. How our immune cells are formed? There is a process called hematopoiesis, which is the formation of red blood cell and white blood cell. Finally, WBCs are the cells of our immune system. So the cells that we study in our immune system are the cells that are WBC, white blood cells. Where it occurs in lymphoid organs. Okay, this process occurs in lymphoid organs. Hematopoietic stem cells, blood, different types of blood plasma, platelets, WBC, RBC, and WBC is our main focused area because these are the cells of immune system. See the classification. So hematopoietic stem cells. Hematopoietic stem cells, remember, they are the multipotent stem cell. Now they can form either myeloid progenitor cells or lymphoid progenitor cells. Myeloid progenitor cells, they form red blood cell, platelets, myeloblast and monoblast. Lymphoid progenitor cell, they form lymphoblast, and lymphoblast develops into B lymphocyte, or you can call it B cell, T cell, natural killer cell. Apart from RBC and platelets, all other cells are the part of WBC. That means myeloblast. Myeloblast again develops into granulocytes, and I think you already know there are 3 different types of granulocytes: neutrophils, basionophils, eosinophils. Monoblast develops into macrophage and dendritic cells. So ultimately, this neutrophil, basionophil, eosinophil, macrophage, dendritic cell, B cell, T cell, natural killer cell, all these cells are the part of WBC. That means our immune cells, or you can call it leukocytes. Apart from this, RBC and platelets. So what you have come to know is that our WBCs, or our immune cells, can be a myeloid progenitor cell or lymphoid also, because see, these cells belong to the myeloid progenitor and these cells belong to lymphoid progenitor. You can't directly say that WBCs are all from the lymphoid progenitor. Indeed, can be myeloid also, lymphoid also, but you have to remember which cells are belonging to the myeloid progenitors and which cell belongs to the lymphoid progenitor. What are myeloid cells? Myeloid cells are the part of innate immune cells, which we'll discuss now. They are the first line of defense and they are present since our birth. Okay, I think you know what is innate immunity. This the immune cells which are present since our birth. Okay, they are the first line of defense. So as you have come to know that the myeloid progenitor cells are the part of innate immune system, and the lymphoid progenitor cells that we have studied, which are the B cells, T cells, NK cells, they are the part of adaptive immunity. So you have to keep in mind which are the myeloid cells, which are the lymphoid cells. Myeloids are the neutrophils, basionophils, macrophages, dendritic cells, and the lymphoid ones are the B cell, T cell, NK cells that you have to keep in mind, that you have to remember. Now, what are innate immunity and adaptive immunity? Innate immunity is the immunity of our body or the immune system which is present since birth. They are present prior to the exposure of antigen. They have a fast action within hours. They are primitive type, means they are the primary one. They are non-specific. Non-specific means they don't have any specific epitope. That means if antigen A and antigen B both have the same, if both have a same type of epitope, then they can bind to either A or either B. That means this innate immunity, it can work on various types of antigens with no specificity. Its recognition is fast. Example: fever, inflammation, phagocytosis, complement pathways. Then physical barrier like skin, eyelid, etc. They can also act like as the innate immunity. Chemical barriers, under chemical barriers, enzymes, body temperature, pH, etc. it comes. So all these things are present since birth and they are like the first line of defense. Cellular barrier, monocytes, dendritic cells, neutrophil, whatever cells that we have studied under the myeloid will be the cellular barrier. Okay. In case of adaptive immunity, what happens? It takes longer times, 5 to 6 days, and it is slower but long lasting. This action is fast, but it is not long lasting, but it is slow, but it is long lasting. It depends upon the B and T cell as discussed before. The response is strong. It is specific to antigen and it forms memory cell. It is highly specific. Adaptive immune system is very specific and it forms memory cells, but here in innate there is no any formation of memory cells. See what happens in innate immunity when the antigen is when the body is exposed to the antigen for the first time, then the response is fast. There is no formation of memory. The response is fast, but it is not long lasting. When the antigen comes to the body for the second time, then the response is also fast, but again there is no memory, the cells development. But in case of adaptive immunity, when the body is exposed to the antigen for the first time, then the response is slow, but here slowly memory cells are also developing. When the body is exposed to the antigen for the second time, then the response becomes fast. Why? Because in the primary exposure, the memory cells are developed. So here, due to the memory cells, the response becomes fast. When antigen comes, then this B cell, it forms plasma cell and memory cell. Plasma cell, it forms antibody, and memory cell, it helps in the secondary response. That means when an antigen infects the body, suppose infects the body, then here B cells will get activated, which is a part of adaptive immune response. B cells will form plasma and memory cells. These plasma cells will make antibodies which will fight with this antigen. And memory cells, what it will do? It will keep all the data of this antibody within it, so that when this antibody will come again in the future, then the action can be a faster. So here it is. In case of adaptive immune response, the response is slow. Okay, within day 10 the response is generated, and these antibodies are formed, and then again when the same antibody will comes for the second time, then the response is higher. See, the response is faster. So this is all about primary and secondary immune response. Next, discuss about antigen presenting cells. Macrophages and dendritic cells are called professional antigen presenting cell. Macrophages and dendritic cells, which are part of the myeloid progenitor, they are called the professional antigen presenting cell. How antigens are presented by this antigen presenting cell? Suppose an antigen entered the body, then what will do? This antigen presenting cell will present these antigens on its surface. Okay, this is the antigen, and this antigen is presented on the surface of the antigen presenting cell with the help of some molecules. This molecule is called MHC, major histocompatibility complex. This antigen is presented to some immune cell. That means this antigen, to whom it is presented? It is presented by the APC, antigen presenting cells, to whom? To cells of the lymphoid system. That means maybe B cell or T cell. And what it will do? It will then destroy the antigen. Now this cell, antigen presenting cell, present the antigen on a surface, which is the MHC. So this MHC can be of two types. One is class 1 MHC or class 2 MHC. So what is MHC? It is a complex on the surface of the antigen presenting cell, which helps to present the antigen to the cells of immune system. It may be B or T cell. So these are the two different types of MHC. MHC class 1 and MHC class 2, there are some structural differences. See here, in case of MHC class 1, there will be one long chain, one short chain. The long chain is the alpha chain, alpha 1, alpha 2, alpha 3. One long chain, which is the alpha, and the short chain is called the beta 2 microglobulin. But in case of MHC class 2, there are two equal length chain, alpha and there are two equal length chain, 2 alpha and 2 beta chain. MHC class 1 is found in any nucleated cell. MHC class 2 is found only in professional APCs. MHC class 1 display endogenous antigen. I have already discussed about endo and exogenous. MHC class 2 display exogenous antigens. MHC class 1 can accommodate shorter peptides. Here they can accommodate longer peptides. MHC class 1 present antigen to CD8 T cell, and here they present antigen to CD4 T cells, two different types of T cell. MHC class 1 has no invariant chain, and MHC class 2 will have invariant chain.